Hugo, thank you, and thank to all the sponsors to help this conference. Of course, we would like to be there in person, but we will have to wait probably another year or so. This is a nice slide here because it depicts a few interesting facts. Here is our champion. This is Jeanne Calment. She lived very long. And I found this picture in circulation research. I wish it was a glass of red wine. I'm sure she drank red wine, which contains high levels of flavonoids. And also you see on the right cocoa beans, and which also contain flavonoids. And we're going to talk about, you know, in general flavonoids, and specifically on cocoa and epicatechin, how this may benefit health. I think during evolution, we probably ate also a plant-based food of high grapes and other types of, you know, vegetables and maybe cocoa. So I think we evolved, and with some of the structures of flavonoids, and our cells may have adapted to some extent to be responsive to them. So I think there's also maybe an evolutionary type of link that these flavonoids actually do something to cells. But again, more research needs to be done. This is a very simplified scheme of why we age. You know, Jeanne Calment, the lady earlier, she was able to escape aging quite well and became very old, 122. But she was still facing many things. And you can see some of the major theories, and we don't need to go into all of these, but we see, for example, DNA damage, telomere erosion, and also mitochondrial dysfunction. But also what you see on this slide is senescence, and this can lead to chronic inflammation. Now there's on the right, you can see there are quite some trials that are ongoing, and we won't be able to talk about NR today, which is also, you know, that's a B vitamin. There's some clinical trials on that we have, and there's many ongoing, but there are trials in epicatechins, resveratrol, and even senolytics, which are compounds to knock out senescent cells. A lot of trials are ongoing there. And you see this little, right here, quercetin is also a flavonoid and is used in these cocktails. So maybe there's something with the structure of these flavonoids that can benefit cells or kill cells as we age and make us, you know, live a little better. This is a complex slide, but the point I want to make here is that although mitochondrial dysfunction is only one mechanism of aging, it links with other of the major theories. In the middle, you see, you know, the oxidative stress can lead maybe through the mitochondria into telomere erosion. We also know if mitochondria gets damaged, this can lead to an increased amount of release of damage-associated molecular patterns, and these can trigger inflammation. So again, the mitochondria is not only an energy organelle that's responsible for energy, but it also has other implications to the theories of aging. As Russ indicated, our goal is to understand and discover biological pathways that cause aging, and then target these in preclinical studies, animal studies, and then, very importantly, help to design clinical trials and also look at the appropriate biological analyses to see if the compound truly affects mitochondrial function, knocks out senescent cells, you name it. In many of the clinical trials, and this is why I'm showing this slide, functional assessments are utilized, such as walking speed. We all like to, improve our walking or our daily activities, or brain function is used, but in a lot of trials, a simple test of walking speed is used, and you can see a paper of, one of all our colleagues, Luigi Ferrucci, that walking speed declines certainly at older age. The next couple slides, I just want to show you that some mutations in the mitochondria are indeed associated with walking speed. And this paper was just published. And we looked at mutations that were specific to the mitochondrial genome and some of the mutations that express some of the complexes. And this was published in Free Radical Biology and Medicine. So we took muscle biopsies of humans with both peripheral artery disease and control humans, meaning healthy humans. We tested functional performance and looked at some of these mutations. I will only show right now some of the data on the healthy control individuals. But what it shows is that 6-minute walking distance is associated with the number of mitochondrial, or the damage. These are strand breaks in ND1 and ND2, normalized to mitochondrial copy number. So again, you can see, strong associations of walking speed and mutations, just to show you that there's an association of maybe, and there's many more papers published on mitochondrial biology and how this is associated with walking performance. So to get directly to the study that was published on cocoa, the question here was whether cocoa, which again is rich in epicatechin, can improve walking speed in older people with peripheral artery disease. On the left here, you see a little bit what this condition is. So these are old people. They have issues with blood flow. There's muscle damage. There's pain with walking, and there's function and mobility. We provided those individuals 6 months with a placebo drink, so no cocoa, or with the cocoa drink. And then we looked at performance. We looked at their ability to walk the 6-minute test. And we also took muscle biopsies. We looked at muscle health, kind of blood flow measures. I'll show you some data and some mitochondrial activity assays. This was a pilot study, so a limited number of people were enrolled in the study. There's a lot of excitement here, eating chocolate to improve muscle health and walking ability in patients with peripheral artery disease. So what does this cocoa do? There are a variety of animal preclinical studies that looked at cocoa, and they gave cocoa to animals. And it improved mitochondrial function, improved growth. Also, there was an indication that it improved nitric oxide, and it reduced the injury in these animals. However, remarkably, the exact mechanisms of epicatechin are really not discovered yet. It's still unclear how this very small structure can have these profound effects. there is some suggestion that it causes eNOS activation. And as you know, nitric oxide, to some, certainly Nobel laureates, they believe nitric oxide is everything. And I would maybe not disagree. It's a very important molecule, and it seems to be increased with epicatechins, and then activates AMPK, and we know AMPK does a lot of good things. But really looking at the literature, there's not much evidence what exactly this small molecule does, and more needs to be done. But I won't be able to talk about that today. So here's the data, and I'll just be 5 minutes on the data and then talk about future directions. So the cocoa was given in a study beverage, and so this was not pure epicatechin, but it was mostly, mostly epicatechin in there and some additional flavonols. A future trial will only use epicatechin, which could be a benefit, also a limitation, because we know a mixture of these flavonols is probably also very, very important. So they received 75 milligrams daily for 6 months. Here again, coming back to a clinical trial structure, there are specific outcomes you need to declare as primary outcomes. You know, cardiovascular function, brain function, or in this case, walking speed. And again, these people have difficulties walking to begin with. And within a 6-month period or a 1-year period, this walking speed actually declines in a typical group like this, because they have these issues. And this is well documented by our collaborators at Northwestern, who these studies are done with. So then there's secondary outcomes like, you know, blood flow. We take muscle biopsies and look for mitochondrial biology markers or senescence, inflammation. Okay, so here's the study design. I'll speed up. So again, this is a pilot study, and at the end, you know, only 44 people were randomized, about half to cocoa and the other half to placebo. You know, clinical trials are not easy to conduct, and this was done with Northwestern, with Mary McDermott, and she's one of the top clinical trialists in the world, and does an outstanding job. So this is the crux of the data. This is the primary outcome. And again, there's very few therapies that work on these people. We've tested a lot of things and nothing helps improve walking speed. So this was the first trial that I've been involved with them where there was a signal. And a signal, we're talking, you know, in this case, 42.6 meters improvement in walking speed, which in these types of trials are very, very significant from a clinical perspective. You can walk 42 meters better than the placebo group. You can see there are 2 points where the walking speed was tested. 2.5 hours, you know, after the last cocoa drink was consumed, and then 24 hours. It is well established that epicatechin and flavonols have an acute effect. So that's why the 2.5 hours. And we wanted to also look at a sustained effect 24 hours later. So is this exciting? You'll see later on that this is exciting enough for a phase 3 clinical trial that will be funded. Am I skeptical? Sure. We got to demonstrate this in you know, a couple hundred people, not in 40. So here are some of the biology. More can be done, or more will be done, but again, a signal on a mitochondrial enzyme that was improved in the cocoa group. So cytochrome C oxidase is one of the complexes in the electron transport chain. Also, you can look at central nuclei in the muscle. If in muscle nuclei are centrally located, this is not very healthy. And you can see that the cocoa attenuated the central nuclei. So there was some indication maybe that the muscle was healthier. We looked and talked about angiogenesis and blood flow. Again, a signal that MRI measures of calf perfusion were improved, a P of 0.11. This is good in the sense that you are going to get more money to do the bigger phase 3 clinical trial. It's not significant, but it's hinting towards it. So in conclusion, cocoa improved 6-minute walk distance at 6 months follow-up. The effects were stronger when measured while the participants was taking cocoa immediately compared to 24 hours later. There were some small changes in muscle biology measures, in COX activity and central nuclei. They were improved. And there was some suggestion on increased perfusion. So what's next? Like I indicated to you, it's very difficult to find therapies for people with walking disabilities. And we just received good news on a phase 3 clinical trial. And there's a 2 percentile, which is a very good score. And this bigger trial will likely start in 2021. So it's a definitive phase 3 multi-centered randomized trial, basically look at cocoa and the effects on walking speed. But I'm excited too, because we're going to look at some biology, and we'll have more tissues maybe for other investigators to look at this as well. At the same time, this past year, I submitted a proposal to the National Institute on Aging Intervention Testing Program. Any of us can submit proposals on compounds that may extend longevity or health span in animals. And this was approved. So basically, different compounds are tested to look at lifespan extension and health span, disease, and disability. So this is done in mice, and this study will start next year. And it's I don't get any money. It's all done at the different universities, and basically epicatechin from Sigma actually will be incorporated into the food of animals, and we'll see if they have a better health span. There is one paper published in FACET that has done some of this in a pilot study, and some of the application proposal was based on that, and they also saw a signal of improvement in the health span. Thank you. I'll stop here.
Great, that was all excellent, Christiaan. While I'm waiting to get some questions from others in the audience, I've got a couple questions for you. So one, in relation to the time points that you looked at, I noted that you had a 2.5 hour post epicatechin ingestion, and then I think it was 24 hours. Yeah, that's the slide. So do you happen to know what the half-life of epicatechins are in the circulation following an ingestion period? So I'm just wondering whether that 24-hour period is close to a full washout.
Yeah, so excellent question. It's not a long half-life. So again, the 24 hours was important because here we're looking for a sustained effect. Let's say if we exercise, we stop exercising for 3 or 4 days, the mitochondria or biology is going to go hopefully not all the way back, but it's going to, you know, some of the effects of exercise are going to be diminished. So yes, there could be acute effects of this compound and then sustained effects. Maybe we should have also measured a week later. And I think in the phase 3 trial, we'll do another muscle biopsy, maybe a month or a month later. I have to go back and look it up to see if there's a sustained effect of the epicatechin.
Okay, great. So a question has popped up in the chat from Dr. Sweeney. Are there plans to test pure epicatechin instead of the cocoa in people?
Excellent question, Dr. Sweeney. So the phase 3 clinical trial that's funded is going to do this with, I don't know, 98% pure epicatechin. So we're going to get this from Mars, the chocolate company. They will generate this and put this in placebo pill. Yeah, so whether that is better than what we've done, we've debated this extensively, because this trial was done in a chocolate cocoa drink, and we also had other flavonols in there, much smaller dosages, you know. But I do think, you know, from a scientific perspective, it's better to do the pure compound and really see if that's doing it.
Okay, great. Dario Coletti has also put in the chat, have you noticed any effects of cocoa flavonoids on neuromuscular function, such as coordination of movement, speed of contraction time, etc.? And I've got a follow-up to that. So
yeah, those were not incorporated in the clinical trial. You know, excellent. I mean, Russ will talk later about neurodegeneration of motor neurons. Yeah, so not done, but could be something interesting to test.
yeah, so just to follow up on that, I was going to ask you if you were planning to look at the abundance of denervated muscle fibers in the patient muscle. And the reason for that is there's quite a bit of data from animal models now showing that recurring ischemia reperfusion episodes cause damage to the motor end plate in muscle. And I believe there's evidence for an accumulation of denervated muscle fibers in PAD patients. It may not be a direct measure, but morphologically.
Yeah, so again, this trial will start next year. If you send us a protocol on how you want the muscle to be removed, and you know, you have to talk to the committee of this trial, we would welcome that.
Okay.
Yeah.